Hi, I'm Rod Green. This series of video clips is going to introduce you to the cavities and the boundaries of the trunk, with particular emphasis on the thorax.
In this first segment, we're going to be looking at the body cavities and considering them as examples of anatomical spaces that you've met previously. We're going to look at the embryonic development of these spaces, and then we'll look at the concept of open versus potential spaces and how they relate to the serous membranes in the body cavities and also to the relevant spaces in the thorax. You've seen all of these principles relating to anatomical spaces previously, so I'm not going to dwell on them at this stage, but the key ideas to remember are that the anatomical spaces have boundaries, and they may be hard or soft tissues, and in terms of functions, the function of a space relates to the structures that are either inside that space or passing through it. The other thing to consider is that there are openings between spaces, and again those openings can be dealt with in the same way as the spaces. They have boundaries and they have functions related to the structures passing through them. So if we go back and have a look at the embryological development now, this is what we call a trilaminar embryo, and this is what we see about 17 days after conception. To visualize what we're looking at here, we could imagine that this flat disc is in fact going to develop into an embryo and then into a person. So we could imagine that this is a person standing up, and we've got a dorsal view of them. So this blue layer, the ectoderm, is the dorsal aspect. And then we're going to cut what would be a horizontal section through that person that's standing up. And when we look at the cut section, we can see that in fact there are three layers. The blue ectoderm layer that we were looking at from the dorsal aspect, the endoderm, which is the inner layer, and the mesoderm, meso meaning middle, so it lies between the outer and inner layers. And you'll get a bit better idea of what I mean by outer and inner when you see the next slide. So these three germ layers are the germ layers that all of our body tissues derive from. If we now concentrate just on the trilaminar embryo, that is this flat disc in here rather than the spaces either side, when we get to the next slide, we can see that in fact that flat disc is now going to start to fold, and that folding forms the rounded body shape that we're more familiar with for either an embryo or the adult body. We can see the same three layers designated with the same colours. So the blue ectoderm is on the dorsal surface, the endoderm is on the ventral surface in yellow, and the red mesoderm lies in between. So as this folds around, we can see that as it's doing that, there's a lot of differentiation of these tissues that you'll talk about at another time, but we can see that as it continues to fold around, the ecto, as the name implies, the outer layer, is going to completely surround the outer surface of this now rounded embryo, whereas the endoderm is going to get pinched off and be completely enclosed inside the embryo. And the mesoderm, we can now see, has split into two layers, one associating with the endoderm and one associating with the ectoderm, and in between we're developing a body cavity, and this body cavity in embryonic terms is called a coelom. On the next slide, we're still dealing with the horizontal section, only a few days later in fact, now 28 days old, but we can see the surface ectoderm now completely surrounds the embryo. We can see that one layer of the mesoderm, the so-called somatic mesoderm, has fused with the ectoderm to form the body wall. The other layer of mesoderm, the splanchnic layer, has fused with the endoderm to form what will eventually become the viscera in the body cavities, and that leaves us with that potential space, the ventral body cavity, or in embryonic terms again, the coelom. And this is now what we're seeing as the generic pattern for the development of these body cavities. If we now related that to what we'd see in an embryo, we're now looking at a lateral view of this embryo a few days later. And so here's the dorsal surface. So this is formed by these areas of mesoderm and so on down on the dorsal surface of the embryo. And the endoderm, again still shown in yellow here, has now started to develop into visceral organs inside these body cavities. So eventually they're going to actually grow out and fill this body cavity. And we're going to look at that idea on the next slide. So firstly, to just summarise in words what we've seen, the somatic mesoderm associated with the surface ectoderm to form the body wall, and the key additional idea to remember now is that because this is somatic, it's innervated by the somatic nervous system. Similarly, the splanchnic mesoderm that combined with the endoderm to form the gut tube or the visceral organs is innervated by the visceral nervous system. And because, as our principle indicates here, the tissue layers of the thorax and the abdomen develop from the same pattern, we're going to have homologous components. And so in fact what we'll see in subsequent video segments is that the coelom, that potential body cavity, is going to form into different named cavities but from the same pattern. So the pericardial and pleural cavities we'll see in the thorax, whereas the peritoneal cavity is seen in the abdominal cavity. To think about these ideas of open versus potential spaces, this analogy with a fist being pushed into a balloon should give you the idea that we're talking about. So instead of this balloon being full of air, let's assume that it's full of serous fluid, which will be secreted by a serous membrane, and we'll talk about these shortly. So as the fist, that is the visceral organ, grows, it's going to push its way into the cavity, and that coelomic cavity that was actually a real space previously now becomes just a potential space in here, as most of the volume is taken up in here by the visceral organ. And the visceral organ now has one layer of serous membrane attached to it, logically named the visceral layer, whereas the outer layer of serous membrane is called the parietal layer, and it's attached on the inside of the body wall.
We've been looking at some theoretical concepts about the development of the body wall and the visceral cavities, and now we're going to have a more realistic look at those on this torso model. So for starters, we can see that the body wall is intact here, so all of the structures on the outside of this torso are all going to be somatic structures. So we see things like bone, cartilage, skeletal muscle, and of course skin would have been covering this as well. In a moment we're going to take the anterior part of the body wall off so that we can see visceral structures as well. Now that we've removed the anterior body wall, we can see the visceral organs inside the cavities. So to remind ourselves again of somatic and visceral structures, we can remember that we had the skin on the surface of the body, then skeletal muscle and bones, so these are all somatic structures innervated by the somatic nervous system. Then inside the body cavity, we have the visceral organs in the thoracic and abdominal cavity that would be innervated by the visceral nervous system. The other thing that we can notice that's quite different on this specimen compared to the PowerPoint slides we were looking at earlier is that in fact the visceral organs completely fill the body cavities. So the coelomic cavity, which was the potential space between the visceral organs and the body wall, has virtually disappeared. So we're going to zoom in and actually have a bit of a closer look at those serous membranes. We can also see inside the body wall here a white line that's indicating a structure attached to the inside of the body wall, and it comes right down here and then continues along the surface of the diaphragm. This is representing one of those serous membranes that we talked about earlier. In this case, because it's attached to the body wall, that would be a parietal serous membrane. You could imagine, it's not really shown on this model, that an organ like the lung would have a visceral surface serous membrane attached to its surface, and those two membranes have the embryological coelomic cavity between them, so it's now very much only a potential space, but they're also important to allow the movement of that organ within the cavity, and we'll see a demonstration shortly that will show that.
We've seen the actual body cavities on the torso model, and we're now going to link that idea back to the serous membrane-lined cavities. So we can see on this coronal section of the trunk, we have the thorax superiorly and the abdominopelvic cavity down here inferiorly. And we'll note that the pleural cavities and the pericardial cavity both lie up here in the thorax, whereas the peritoneal cavity lies lower down here in the abdominopelvic cavity. If we look at one of those in detail, and we're going to concentrate on the thorax because that's your main area of study, we're going to look at the pleural cavities that surround the lungs. So we've talked previously about the fact that these derive from the embryonic coelomic cavity, and there are two layers of pleura. So the visceral layer, represented by this orange line, surrounds the lung itself, which is shown here in cross section, and the multicoloured line lying on the inside of the body cavity is the parietal pleura. So we can see that most of the body cavity is actually filled with the visceral organ, and only a very small potential space remains, and in this case, because it surrounds the lungs, that potential space is called the pleural cavity. So these serous filled cavities lie within the major body cavities, and their main function is to actually reduce friction between the organs and the body wall, and this allows the lung, as we breathe, to move relatively freely inside the thorax. If we now come back to the idea of open versus potential spaces, the pulmonary cavity is the actual open space that lies in the thorax on either side of the mediastinum, and it contains both the lung and the pleural membranes, and its boundaries are the body wall itself, and its internal surface is lined with the parietal layer of pleura. In contrast, the pleural cavity is the potential space that normally only contains a very small volume of serous fluid, and its boundaries are the parietal and visceral pleura. Very briefly, these principles will apply to all of the cavities, but we've seen an example that the body cavity can contain hard or soft tissue walls, and they're generally lined with secretory membranes, in this case the parietal pleura. These body cavities contain organs, in this case the lung, and these organs have related functions, and the fact that they're in separate cavities can potentially limit the spread of infection between compartments. And the key idea is that the serous fluid-filled cavities, that is the potential spaces, the pleural cavity, are going to allow the movement of the organs that project into them. So what we in fact have now up in the thorax is that the pulmonary cavity, the open space, contains both the lung and the pleural cavities, and the middle mediastinum contains the heart and the pericardial cavity.
So we've talked about the visceral serous membranes lying on the surface of the lung, and I've got two sheets of laminated paper here that are representing these membranes. So this sheet is representing the visceral serous membrane, and I've then got a second sheet that's representing the parietal surface membrane that's attached inside of the body wall, represented by that white line. If we now zoom in on these sheets, you'll get the idea that I'm thinking about. Now that we've zoomed in on these laminated sheets, you can see the words that I have written here. So a visceral serous membrane is always attached to the surface of a visceral organ. So in this case, we could take the example of visceral pleura that's attached to the surface of the lung. The second laminated sheet that I have on the other side represents a parietal serous membrane that's always attached to the somatic structures, the body wall. So for example, if this was parietal pleura, it's attached to the inside of the thoracic wall. And the important concept to understand here is that if I put a hand on each of these so that they move independently, as the lung expands and recoils during respiration, it's going to slide easily over the thoracic wall, and the very thin layer of serous fluid that lies in between these serous membranes facilitates that easy movement.
To ensure that you've understood the content of this video segment, I'm going to ask you to complete a task. What we have here is a horizontal section through the trunk, and on that section we've got three organs that I've highlighted: organ A, organ B, and organ C. And I'm going to ask you on the next slide to complete a series of questions related to those three organs. The key idea to complete this task is to remember that in cutting through the trunk with a horizontal section, I've cut through a number of body cavities. For each of those three organs, you're going to answer the same three questions. Firstly, you're going to name the organ. Then you need to tell me what's attached to the surface of that organ. And lastly, I want you to tell me which cavity that organ lies within. Once you've attempted those questions for each organ, I want you to post your answers on the relevant discussion forum.